So now I'm going to try to summarize some of the more recent single-cell RNA-seq based technologies that have been developed over the past few years.
So this slide gives a pretty incomplete now overview of different single-cell RNA-seq based technologies. And so the main distinction between the ones listed here anyways are whether or not the particular technology sequences full-length transcripts or only does 3' taking. And so methods that assay full-length transcripts basically generate reads that can come from, in theory, any part of any transcript. And so they typically involve some kind of like fragmentation of full-length transcripts into different smaller fragments, which you then get sequenced, analogously to the bulk RNA-seq protocol we talked about earlier. And in contrast, the 3' taking methods really only focus on sequencing a small short read out of the 3' end of transcripts. And so you can see in this table below that there's basically a kind of a inverse relationship between the amount of detail you get out of an individual transcript versus the throughput of the assay. So what I mean by that is if you look at the full-length based approaches, generally speaking, the throughput or the number of cells that you can sequence is typically somewhere on the order of at most like 100 or maybe say 1,000 cells per assay. Whereas when you look at the 3' end counting methods, they really allow you to sequence a few orders of magnitude more cells compared to the full-length approaches. And so, for example, split-seq, at least according to this table, allows you to sequence somewhere between like 1,000 and 10,000 cells. And that number is actually much higher nowadays. So you can actually use, for example, Chromium based approaches to sequence like hundreds of thousands of cells fairly easily. And so really, but if you look at the typical read depth per cell, the full-length approaches tend to give you like a higher number of reads per cell compared to the 3' end counting methods. And so really the choice of full-length versus 3' taking methods is a choice of how much information you want per cell. So typically the full-length transcripts approaches allow you to get more reads per cell, and they also in some sense give you more information per cell or per transcript, because since your reads come from all over any given transcript in principle, that means that you can detect, for example, alternative splicing that occurs early on in the transcript. Whereas for the 3' taking methods, because you're only sequencing a small portion of the transcript at the 3' end, that means that you don't see what happens in the 5' end. And so there actually are now protocols for doing 5' taking as well, but they're not discussed here. And they would also suffer from the similar problem that if you only sequence one end, then you don't know what happens at the other end. And so another really important point I want to make here is that, again, the throughput that we're talking about here is on the order of like hundreds to like hundreds of thousands of cells. And so in comparison, some of the biggest gene expression based studies that used bulk RNA sequencing methods, such as the GTEC project that sequences different human tissues, even these kind of super large consortium that did bulk RNA-seq assays could only really sequence on the order of hundreds or now kind of low thousands of samples for the entire project. Whereas with the single cell based assays, you can really sequence like 100,000 cells all in one experiment. And so the scale of the data that comes out of the single cell based assays is just orders of magnitude more than bulk samples. So the Drop-seq single-cell RNA platform, which was later commercialized by 10x into their Chromium product, is one of the most high throughput, frequently used platforms for doing single-cell RNA-seq. And so we'll briefly discuss how it is used and how it works here. And so the idea is that you start out with a microfluidics device that basically has 3 input channels shown on the left. One that's used to pass in dissociated cells, another for microparticles and lysis buffers, and a third for oil. And so we'll discuss what the microparticles or beads are used for on the next slide. But basically, when you add the lysis buffer in with the oil in the cells at the right concentration and speed, this basically causes droplets to form, where on average, you hopefully get one bead in one cell within each droplet. Although sometimes you'll end up with empty droplets, or sometimes you'll end up with droplets with a bead and no cell. Or, for example, you'll get a droplet with a cell but no bead. And so the idea is that after the droplets are formed, the cells get lysed, and the mRNA contents get dumped into the droplet. And so the idea of the beads is that each one of these beads, or what are called microparticles here, have a bunch of primers hanging off of them. And each primer has a poly-T tail that basically allow them to get hybridized to the poly-A tails of mRNA from the lysed cells. And so the key idea here is that, and we'll show this later on the next slide, is that for every bead, all of the primers hanging off of that bead have a unique cell barcode, and it has the same cell barcode on each bead. And the idea is that this cell barcode basically allows you to later on identify that all the mRNA that hybridized to that bead came from the same cell. And so again, every primer hanging off of a bead has the same cell barcode. But on the other hand, as we'll again see later, every primer also has what's called a UMI, or unique molecular identifier, where hopefully every primer has its own unique UMI for a given bead. And so after the mRNA hybridizes to the poly-T primers on the beads, then the droplets get broken and the microparticles get collected and washed. And so here the important point is that even though the droplets are broken and therefore all of the different beads are pulled together, because each of the beads has its own unique cell barcode and all of the primers on that bead have the same cell barcode, then all of the mRNA that get hybridized to a single bead have to come from the same cell. And so the mRNAs then get reverse transcribed in bulk, and that basically forms what are called stems. And then a technique called template switching, which we won't go over here, is basically used to introduce a PCR adapter downstream of the synthesized cDNA. And so the cDNA get released from the beads, and then they get PCR amplified. And as we'll talk about on the next slide, the PCR amplification process is a bit more robust than the one typically used for bulk RNA sequencing. And because of the unique molecular identifiers, or UMIs, we can basically, in large part, address the PCR amplification bias that plagues a lot of bulk RNA sequencing assays. And so after we sequence the reads, then as usual, we can then map them to the genome or the transcriptome, and then identify which gene each cDNA came from. And so again, because each primer on the bead has the same cell barcode, that means for every cDNA that we sequence, we know which other cDNAs came from the same cell, and so we know their cell of origin. So the 10x Chromium approach is called the 3' tagging approach because these essentially full-length cDNAs are sequenced using, for example, paired-end short read sequencing. And so what happens is that on one end, you're going to sequence some barcodes that tell you the cell of origin. And on the other end, you're going to sequence part of the 3' end of the transcript. And so using that 3' end, that's how you're basically going to identify which gene the transcript came from. But what that means also is that because you only sequence the 3' end of the transcript, then you may be able to identify the gene that the cDNA came from, but you won't be able to identify, for example, alternative splicing events that happened in the 5' end that are past the barcode that you sequence. So part B of this diagram shows you an actual illustration of the microfluidic device used in Drop-seq. And so here you can see the beads basically come in through the central channel moving left to right. The cells come in from two channels, the top and the bottom, and oil basically comes in through the top. And basically the droplets form and move left to right as they get lysed, and then the mRNA gets hybridized, and so on and so on. And so part C of the diagram basically illustrates a single cDNA molecule constructed in this library. And so you can see that basically the reads labeled read 1 and read 2 represent a hypothetical paired-end sequencing of the single cDNA molecule. And you can see that basically the read corresponding to the 5' end of the cDNA captures both the cell barcode as well as the UMI, and the read coming from the 3' end of the transcript basically captures the 3' end of the transcript and is used to basically identify the gene that the transcript came from. And so part D illustrates a hypothetical library that corresponds to a whole bunch of cDNA sequenced from a given single-cell RNA-seq experiment. And so the important point to note here is that this is an example of the hypothetical library that you might generate, say for example, before PCR amplification. And so how we get around the PCR amplification bias is that when PCR is applied to increase the total number of copies of each cDNA, what you'll note is that every original cDNA molecule has a unique molecular identifier that tells you that corresponds to exactly one transcript in theory that was hybridized to one of the beads. And so even if certain cDNA molecules get amplified more often than others, you'll be able to tell what that amplification bias is, because if, say, the first cDNA molecule was amplified 10 times more frequently than the second one, you'll actually see the UMI barcode for the first cDNA molecule get sequenced 10 times more than the second one. But you can actually tell how many times each cDNA molecule was amplified, relatively speaking, because you'll just see the same UMI get duplicated over and over and over again. And so you can actually tell how many original transcript molecules there were, because that will correspond to the number of unique UMIs that you sequence in your library. And so again, after you align the cDNA to the genome or to a reference transcriptome, and you group them based on, you group cDNAs that have the same cell barcode together, because you know that they came from the same cell. And that allows you to essentially count how many times each gene is represented within each cell in your library. And that's how you get your table that represents your single-cell RNA-seq experiment that tells you how many times each gene was basically identified in each cell. So this figure is from a study that measured the performance of a large number of single-cell RNA-seq protocols and compared them basically based on their ability to quantify the expression of spike in RNAs that correspond to essentially fake genes of known concentration. And so they basically defined accuracy here in the top figure as how close the estimated relative abundance levels of these fake genes were to the actual known abundance levels of these spiked in molecules. And so high accuracy basically means that they captured the relative abundance correctly, and therefore suggests that the variation in expression that you measure with that technology is closely corresponding to the actual variation that occurs within these cells of the sample that you sequence. And so this is as opposed to the variation being driven by, for example, technical factors that are not related to the underlying biology. And so here you can see that across the different single-cell RNA-seq methods, there's a huge variation in terms of the accuracy across, not only across the different methods, but within a given protocol, you can see a huge variation. So each point here represents a single cell. And so, for example, for some technologies like MARS-seq, you can see wide variation in terms of the correlation between the abundance estimates and the actual known concentrations. And so you can also see that, for example, bulk RNA sequencing methods, generally speaking, give you a relatively high accuracy per profile estimated in comparison to a lot of the single cell methods that do show a lot of variability. And so the second metric that they also measured is what's called sensitivity, or what's precisely defined here as the molecular detection limit. And so the molecular detection limit is defined as the minimum number of input RNA molecules that you need in order to confidently detect a gene, or in this case, like a fake gene. And so higher sensitivity, or essentially a lower detection limit, means that you can detect very weakly expressed genes. And so you can see here that bulk RNA sequencing actually has relatively poor sensitivity, or correspondingly a high molecular detection limit. So that means that you need to sequence a lot of molecules, a lot of RNA, in order to detect weakly expressed genes. And so in contrast, most of the single-cell RNA-seq methods have a lower molecular detection limit. But again, you can see that even across the protocols, or even within a given protocol, you can see that there's a huge variation in terms of the molecular detection limit.